Leading a startup team, whether you're delivering a sugar rush, stocking coffee, or getting a regular delivery of snacks, Office Depot has solutions that fit every startup culture, from getting those first business cards and stationery to ordering fleece pullovers with your new logo. To learn how Office Depot and the California Technology Council have partnered to bring you savings on all of these startup essentials and more, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report. There's been much made of the potential of the microbiome to address disease and promote wellness. While much of the therapeutic efforts in this area have focused on the microbiome of the gut, Azitra has developed a platform for selecting bacteria native to the skin and engineering it to produce therapeutic proteins. We spoke to Travis Whitfill, Chief Science Officer of Azitra, about the company's platform, why it may be preferable to apply bacteria to the skin that can produce therapeutic proteins where they're needed, and why some of the biggest opportunities for the technology may be in the health and beauty markets. Travis, thanks for joining us.
No problem. Thanks for having me.
We're going to talk about the microbiome, Azitra, and how you're harnessing the microbiome to treat a range of skin conditions. I suspect many listeners have heard of the microbiome, but I think there's probably a tendency to think of it in terms of the gut and as potential treatments for gastrointestinal diseases. What does the skin microbiome consist of, and what role does it play in the health of the skin?
Sure. So, as I'm sure you know, the skin is the largest organ of the body, and it has an incredible surface area and interacts with the environment and kind of serves as the primary barrier between you and the outside world. So, in fact, there's a ton of microbes on the skin. There's about 1 million per square centimeter, and a lot of times, when you think of the microbiome, you think of gut health, you think of yogurt, you think of oral probiotics, but in fact, there's a really large role of the skin microbiome health. So it's a large interface between the person and the environment. So it's about 1 million microbes per square centimeter of the skin. Very diverse communities consisting of bacteria, fungi, and viruses. Mostly bacteria, but there's quite a few of the other players. And it's really kind of variable by skin type. There's classifications based on moist skin, dry skin, and oily skin, and each of those different classes of skin have their own microenvironment and diverse microbes. And each skin site differs between individuals. So there's a lot of diversity and variability between people. And as soon as you have a disruption in the skin microbiome that doesn't necessarily cause a disease, but it's associated with a number of different diseases, especially eczema, atopic dermatitis, rosacea, and a couple other skin diseases that are pretty common. So a really big interplay between the skin microbiome and health in general.
Azitra has developed a microbiome platform for selecting an ideal strain for an indication and then engineering it to deliver a therapeutic protein. How does your platform work?
Sure. So the general idea that we had is that we can use microbes to deliver therapeutically relevant proteins to the skin. Because that way you have a microbe that's delivering a constant dose of a protein. It's getting deeper into the skin, and it's delivering it constantly over time in situ, basically. So that's the broad idea and the goal of the platform we envisioned. And we're using Staph epidermidis as our host. and kind of the basis for this platform. And staph as we call it, is a very common skin commensal. It's found on everyone's skin, and it's one of the more common microbes. And there's been a lot of really good research recently about the role of staph in the skin, especially its anti-inflammatory effects and kind of maintaining homeostasis on the skin. So a lot of really good properties of staph. And we've engineered it to secrete a number of different human proteins that could be relevant for skin disease. We have this vector that we've engineered in this plasmid. We can insert any gene of interest that we can imagine that's, kind of feasible to put in the bacteria to secrete a number of different proteins. This is especially relevant not only in common skin diseases that you can secrete a structural protein or an anti-inflammatory cytokine, but also in rare skin diseases where you have, a certain protein that's missing that's causing a skin disease, or, missing that is, causing severe reaction, things like that. So, you can think of it anywhere from, delivering a therapy to protein replacement, things like that. So that's kind of the overall goal of the platform, and we've developed specific iterations of that that can go into more detail.
Well, when I think of the microbiome to treat a condition, I think of harnessing a bacterial strain or a colony of bacteria to restore something that's missing or interfering with part of a disease process. You seem to be using it more as a delivery system and, in some sense, a manufacturing system to produce a needed protein where it's needed. Is that correct?
Yeah, exactly. So, instead of delivering a topical, we can have the bacteria make it in situ. So we have, basically the bacteria themselves are the protein factory. It produces a reliable and stable amount of protein over time. It's always making protein. And we've engineered our system so that there's a secretion signal on the protein so that the staph can excrete it or secrete it right as it's being made. So that way it gets out of the cell, kind of reduces the metabolic burden, I guess, on the cell, and gets into the skin. And then we've engineered another part of the protein that helps deliver through the stratum corneum and more deeply into the skin tissue to kind of deliver, more to the site of action.
Why is this preferable to, say, producing the therapeutic protein itself and just delivering that directly to the skin?
Sure. That's a really common question that we get all the time, and sometimes we ask ourselves that. But essentially, first of all, purifying protein in manufacturing is not cheap. It's very expensive to purify protein, especially if you're working with mammalian cells or even yeast. It gets really expensive quickly, and you get a pretty low yield. Whereas our system, if you're just manufacturing bacteria, it's very cheap, and we've been able to show this with our manufacturer in Europe about the cost of making the bacterial strains itself, and it's a really cheap solution. First of all, second, we think that it would be more efficacious because instead of delivering a single dose of protein to the skin that would then get degraded, the bacteria would make the proteins and continually be expressed and secreted over time. So that way, you only apply the bacteria once, but throughout the day, they're making the protein locally in the skin. And that way, you don't have to have repeat applications. Because a lot of times, the skin has proteases that can interfere with proteins, can degrade them really quickly. Whereas this, it makes it at the site of action and continually does so. So those two questions. Additionally, there's kind of a synergistic effect with staph in addition to the protein. So staph, as I kind of alluded to earlier, has a lot of really beneficial properties. There's been a lot of incredible work at the NIH by Yasmin Belkade and others, Trudy Knight, who's a postdoc, in our lab did a lot of really cool research on Staph epi. They showed that it kind of bolsters the skin immune system. There have been other labs like Rich Gallo that have shown Staph epi isolates that can kill Staph aureus, and there have been others that have shown that it can help accelerate wound healing in animal models. Even there's a human clinical study in Japan that showed that Staph epi can help improve hydration in the skin, reduce water loss, etc. So, all of those properties are really beneficial that we're kind of adding into our system. And then with the added benefit of protein replacement and protein delivery. So we see it primarily as a synergistic effect, improved over just delivering protein plus, the cost of goods and manufacturing issues I mentioned.
And do you know how long after it's applied to the skin it lives and continues to produce the therapeutic protein?
Sure, yeah. So we've done a lot of work in kind of, building in some tools that can control that time span. So, we do have the ability to make something that can colonize the skin forever. That might be nice, but in talking with the FDA and other people, from a safety perspective, we've tried to kind of limit the lifespan. And so we've engineered our bacteria that are actually oxytrophic, so that we've knocked out a couple of genes in the genome of the Staph epi so that it doesn't survive without the addition of our plasmid. And so as soon as the bugs kick out the plasmid, which is in about 1 to 2 days, then the bacteria would basically die, and we do have to be replaced. So we've tried to kind of engineer it and tune it so that it's a daily application, so that way, the bacteria would make the protein over time and then start to die off about a day, and then you replace it. If that makes sense.
I take it also the fact that this is a much lower cost manufacturing process opens up other markets to you, such as cosmetics and beauty supplies, where you may have benefit, but the costs are different than, say, producing a biologic.
Absolutely, yeah. I mean, it's not quite as cheap as making yogurt, but it's not too far off. So, we are talking with a number of different potential cosmetic companies that are interested in this. A lot of skin health applications, especially in dry skin. So, that would be with a non-modified version of our Staph epi that wouldn't be secreting a human protein. But there's a lot of potential plays there with consumer health and over-the-counter products, things like that. So it's definitely an area we're exploring.
Your lead therapeutic, AZT01, is in development to treat eczema and ichthyosis vulgaris, a condition where the skin doesn't shed dead cells. What exactly is AZT01 and what does it do?
Sure. So I'll kind of start with what is eczema and ichthyosis vulgaris. So eczema is a very common condition that a lot of people have, and it's caused by, I think a lot of people know what it is, but it's essentially, red skin, inflamed skin, a lot of patches, really severe in childhood. Most people kind of grow out of it, so to speak, but 10% of children with eczema carry it into adulthood. Pretty severe disease. The treatment options are pretty poor. Calcineurin inhibitors, steroids, things like that. They have black box warnings from the FDA, some safety concerns with those. So there's a lot of room for innovation in eczema. And it's caused by three major components, where you have a structural protein, structural defect in the skin, often caused by a lack of filaggrin expression. And filaggrin is the protein that we've engineered our Staph epi to express for AZT01. And filaggrin helps with binding keratin filaments to the skin and sealing off the skin to the outside world. So it helps retain skin moisture, helps protect against antigens and pathogens. So when you don't have filaggrin, you can have destructuring of the skin, leads to potential antigens coming in, and then in eczema specifically, you have inflammation driven by a Th2 response, and unique to eczema, you often have flares driven by Staph aureus. So Staph aureus is a really common bug in eczema, really contributes to the disease severity, things like that. So that's eczema. And ichthyosis vulgaris is a pretty unique disease where it's caused by filaggrin mutations. 100% of people with this disease essentially have a filaggrin mutation, and it's characterized by dry, scaly skin. Ichthyosis is the Greek term, I think, for fish. So it's scaly skin is the hallmark of it. So in both cases, we're developing AZ201 for these two indications where it's Staph epi that's secreting filaggrin. And filaggrin is a pretty complex protein with 12 different monomers, but we've selected one that we think makes sense based on some structural activity relationship studies and other studies that we've done in our labs. We've engineered our Staph epi to secrete it. So the idea is that basically in ichthyosis vulgaris, it's a protein replacement therapy, and then in eczema, you have the combined effects of the anti-inflammatory effects of Staph epi combined with filaggrin helping reestablish the skin barrier.
What's the development path forward?
So we're currently in preclinical studies. We're talking with the FDA actively and working with our regulatory consultants to kind of finalize the IND investigational new drug application that we would submit to the FDA, and we would do several clinical trials in 2019. So we're hoping after meeting with the FDA and securing the path forward from what they want to see in terms of safety and tox studies in the animals, then we would file an IND with the FDA and start our clinical studies next year. So the initial phase 1 study would be in healthy volunteers, followed quickly by a phase 1B study in either or both ichthyosis vulgaris and eczema patients. And we're always exploring potential partnerships with other pharmaceutical companies that would be able to take it forward after we're able to demonstrate efficacy. But first is safety and then efficacy in these human studies.
You have another product candidate that's in development as a consumer product for rough, dry skin. This is actually the potentially largest market you're addressing in your pipeline. Is the expectation this would be sold over the counter, and will it need to go through a therapeutic development path?
Yeah, so my background is kind of in drug development, so I've been learning a lot about consumer health and the cosmetic markets and the regulatory path for that. So for this, our candidate would be the base strain of Staph epi. That's an oxytrophic strain that wouldn't be secreting any proteins. And so that way, it's clearly differentiated between the therapeutic strains that we're developing that express human proteins, and then this is the non-therapeutic strain that would be developed for consumer health. And the difference really, from a really strict perspective is the claims that you make in the product. So for anything that's in the consumer health market, you can't make any health claims. You can't say that it treats any type of skin, but we're able to make general statements and monograms is what they call it. So, helps address the signs and symptoms of dry skin. So that would be, the claim or the statement that we would use for the consumer health products. And those are not regulated by the FDA. They are, but they don't require pre-market approval.
They don't have to go through a clinical trial process.
They do, and we will, put them on humans before you commercialize it. So we will have some clinical studies, but it goes through an IRB process and not have to be approved by the FDA. So that's really the key difference there.
And how might that product be preferable to what's on the market today?
Sure. I think that a lot of consumers are starting to be aware of the microbiome, and some of these consumer products damage the microbiome and aren't really microbiome friendly. A lot of them have pretty harsh chemicals or additives. This would be different in that, it's compatible with the microbiome. You don't have to disrupt it. It can colonize your skin, and it's natural, safe, and it's a different effect that we're seeing. Instead of a moisturizer, it's kind of restoring the balance of your microbiome type idea.
As a living product, are there any unusual challenges from either a regulatory perspective or from the way this product would ultimately be stored and dispensed?
I would say that, I guess unusual, yes. Of course, living organisms as a therapy or as a product is definitely, a little more on the unique side. But in terms of the therapeutic aspect and the regulatory aspect for FDA-approved products, there's a very clearly defined path forward, and it's called a live biotherapeutic product, or an LBP. And that path is very well defined from the FDA. They've issued guidances. They know what to expect, and they, have, kind of all of the manufacturing expectations set. So it's a pretty solidified path at this point, even though it's pretty novel, and there are few or none approved live biotherapeutic products on the market. There are a lot of trials going on, and the FDA has issued a lot of guidance and input on this. So from the regulatory perspective, we feel pretty good about it. From the commercialization perspective, the hardest challenge is going to be what the consumers will want, and convincing them that there's need and efficacy with this product. But in doing a lot of the market research that we've conducted so far, we really feel comfortable and confident that consumers want this product, this type of product. They need something different for their skin, especially for the pharmaceutical indications. So we feel confident that there's a path forward, both from the market need, from the FDA's perspective, and we've already worked out the manufacturing challenges. Like I said, the cost of goods is really low, and we've been working with a wonderful partner in Europe that's helped us through that. So, comprehensively, this is a new area for sure, but we see a really well-defined path forward, and we're kind of trailblazing a little bit here, but feel confident about where we're headed.
Travis Whitfill is Chief Scientific Officer of Azitra. Travis, thanks so much for your time.
Thank you. Great to talk to you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.